Okay, welcome back, everybody, for our second session of the afternoon. We have a terrific lineup here. Our first speaker is Michael McCormick, who is a professor of medieval history at Harvard, and he chairs the new university-wide initiative for the science of the human past at Harvard. That's an interdisciplinary research network that brings together geneticists, archaeological scientists, climatologists, environmental, computer, and information scientists, humanists, and social scientists in order to explore the great questions of human history, from our origins in Africa to our migrations across the globe. His current research interests focus on developing new archaeological, scientific, and textual approaches to the fall of the Roman Empire. He was elected to the APS in 2003, and he's going to speak to us today on the new science of the human past.
Thank you, Linda. And thank you all for being here. It's a great joy to be with you. I'm only sorry that we can't be together physically, because you will always fill me with questions and ideas and suggestions days and hours after the talks as we had our coffee and so forth. But I hope you remember a little bit. And when we're together again physically, I'll have that privilege. Over 50,000 years ago, when some of us left Africa for the first time, we were already asking ourselves, who are we? Where do we come from? How did we get here? The answers to those questions have been largely myths until about 200 years ago, when the first of three revolutions occurred in our knowledge of the human past. Around 1800, historians invented the seminar system and teamwork that reconstructed the past, not from the claims of politicians and their spinmeisters and biographies, but directly from the archival documents produced in the time itself. Around 1900, archaeologists invented excavations to extract physical pieces of the human past from the soil and from the sea, and to use them to reconstruct what happened in places where the text ran thinner. In our own time, scientists and scholars are inventing a new science of the human past, using ancient biomolecules to reconstruct the invisible remains of the ancient world that physically are present among us. These are the goals of the science of the human past at Harvard: to bring the power of 21st century of science to shed light on the human past as it happened in its splendor and misery over the millennia, to create new knowledge of the past that informs the present, to engage students at all levels in creating the new knowledge, and to create a critical mass of researchers who speak the language of history and science alike, and so to bridge the divide between the two cultures of science and the humanities that have been separated since the 19th century. Our initiative is a network of seminars and laboratories and research groups, and is guided by a steering committee recruited from half a dozen departments across the university in as many disciplines in 3 different divisions of the university. In some sense, the science of the human past springs from archaeology. When you excavate and remove material pieces of the past from the soil, you hold pieces of the past and are able to bring to bear on them all the power of modern technology and science to extract the invisible information they contain about the people who preceded us. I want to highlight 4 different areas of investigation in our initiative at the present time. I'll pass rather quickly over archaeology and data science and offer a little bit more depth about environmental science and what we're doing in the world of ancient DNA and ancient biomolecules. In archaeology, we have a number of international collaborations, most notably in recovering through minimally destructive technology the mysterious site of a Visigothic capital founded by invaders of the Roman Empire in the 6th century, the only Roman-style city supposedly founded by the Germanic newcomers. We don't have the resources to excavate. Much of the land is in private property, so we have to investigate without digging. And so we use various technologies, including remote prospection, to detect structures that are invisible today but are present underneath the soil. Here you can see circled everything that had been excavated in 75 years of study at the site about 150 kilometers southeast of Madrid. That was a palace. Was there a city there, as the chroniclers claimed? There was no sign of one. In 2 weeks of work with my students and our colleagues from Germany, we were able to map with geomagnetic devices the structures that lay underneath the soil and uncover streets, houses, and so forth. The skeptics, like Mike McCormick, were wrong. In 578 CE, a Visigothic king did indeed found a city at this site of Reconstruction. Here you can see the drawings of what was found. The yellow area was what was known before the geomagnetic campaigns that we began in 2015, and everything else is new. This little structure in the corner is a completely different orientation and design from all other buildings. This site was a Visigothic town, and then when Spain was conquered by the Arabs in 711, survived for a few decades as an Islamic center. We believe it is possible, but of course we can't excavate, that this is the oldest mosque surviving in Europe. Data science is transforming our knowledge of the ancient world through the ancient texts, which are now increasingly available in online versions, which allow us to do gigantic searches for word occurrences, multiplying many times the evidence, the philological textual evidence we can bring to bear on great questions, and by applying quantitative and other methods, allow us to identify hitherto unidentified texts, date them and localize them, and acquire amazing new written sources for antiquity. Another aspect of data science and the science of the human past is the digital atlas of Roman and medieval civilizations, soon to appear in a new version of maps, which applies geospatial technology to map the ancient world. It's been created over more than a decade largely by Harvard undergraduates, and we have been mapping the ancient world, putting it up in an online free atlas. Go check it out. 3 different groups of economists have used our freely downloadable data sets of Roman roads in their investigation of what is the single most powerful variable to explain economic vibrancy in different regions of Europe today in 2021. The answer, the proximity, the presence of Roman roads. Roman roads. That's some bang for your 2,000-year-old infrastructure buck. The third involves environmental science, and one of the gems of the science of the human past is the Historical Ice Core Project, another international collaboration which successfully retrieved a core from the Swiss Alps at Colli on the Italian border. I co-direct this with Paul Majewski, the eminent climate scientist and glaciochemist at the Climate Change Institute of the University of Maine. Paul's team has developed an amazing new laser ablation instrument, which increases manyfold the resolution of our analysis of ancient air trapped in ice levels, in glaciers that we take out from ice cores. The conventional technology used until now is called continuous flow analysis. It requires you to destroy the ice by melting it. Its finest resolution is 1 centimeter. That's fine if you're working in Greenland or Antarctica, where the ice cores down to the beginning are 3 kilometers long. Our ice core is 72 meters from 2013 to before 2,000 years ago. That means that the ice, which contains the ancient air, is highly compressed, and the divisions, the distinctions are invisible to the conventional technology, but they emerge clearly with the laser ablation inductively coupled mass spectrometry with the cryo chamber that Paul Majewski's team has developed. And so you can see here different studies that have emerged in the last few years at different levels of the ice core. I'm just going to highlight a couple for you of particular saliency. Governments and non-GOs have agreed for decades that atmospheric lead pollution has been a terrible problem since the beginning of the Industrial Revolution. Our measurement of atmospheric lead pollution deposited on the top of the Alps has demonstrated that it's not been a bad problem for the last 200 years, but at least the last 1,000 years. You can see right here that around 1200, the levels of lead pollution are completely comparable to those of the early Industrial Revolution, indeed of the 19th century, with one exception, that drop right there, 1349 to 1354. That's the only time in the last 2,000 years that atmospheric lead pollution is undetectable on the surface of the Alps on the Swiss-Italian border. Why is that? Because between 1349 and 1354, the bubonic plague in the form of the Black Death killed the miners, stopped mining and metal production, and abated lead pollution back to natural levels for the only time in the last 1,000 years. The inset shows the ultra-high resolution of our lead measurements, and the gray histogram shows the archaeological and historical evidence that shows the closing of each mine year by year, month by month, as the plague spread. The rose-colored ones are the archaeologically and historically documented reopening of mines. And you can see that the fit is pretty darn good. Some of you may remember a movie and play from a few decades ago about Eleanor of Aquitaine, Queen Eleanor of Aquitaine, and her umpteenth husband, King Henry II. Our Ice Core delivered some amazing new insights to their reign. And they involve the lead produced at some of the most important royal mines of England, here at Peveril Castle in the Peak District at Castleton, the castle that was built in the 1170s to protect these mines of King Henry II, since Eleanor had disrupted them with a little war and a tiff that had sprung out between the two. On the top, you see the measurements of lead deposits in the 12th and early 13th century on the device at the Climate Change Institute in Maine. And on the bottom, the tabulation of cartloads of lead delivered, sometimes a year or more after they were removed and refined, to the Royal Treasury in London. The connection between the Royal Archives and the chemical signal in the Alps is remarkable. In 1170, Henry II ordered the murder of Thomas à Becket. The Pope responded by throwing the kingdom under interdict. Everything stopped. And you can see that there's a drop in the atmospheric pollution, and deliveries to the Royal Treasury of recently mined and refined lead stopped precipitously. Over and over again, whether it's a war or departure, when the king leaves for crusades, they stop collecting taxes. When there's a transition, when Henry II dies and Richard Lionheart takes over, they stop collecting the taxes. So you can see over and over again this remarkable connection between atmospheric lead pollution measured on the top of the Alps and the archival records preserved in those scrolls that you see in the corner of the slide from the 12th century monarchy of Britain. Wonderful confirmation of the power of science to uncover the economic development of the past. And we published that just last year in this article in Antiquity. A sister organization here in Cambridge has been issued quite recently a report on the role of international science in US science, and it emphasizes that US science will not be able to compete globally unless it goes globally. It grows continuously. Science is international. It is a collaborative effort if it is to be successful. This is embodied by our new virtual research center sponsored by the Science of the Human Past, the Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean, which many people pronounce MOM and which I occasionally pronounce mayhem. It's a virtual research center that connects the complementary strengths of the laboratories of the Max Planck Institutes of Jena and Leipzig with those of Harvard University. Our leadership team of directors and deputy directors is international, drawn from both institutions. And I've been able to raise the funds for 3 new graduate fellowships at Harvard. We have 3 graduate students being trained simultaneously in the humanistic and social sciences of history and archaeology, and the sciences of isotope studies and genetics, so that the future generation will come of age being able to speak both the languages of the humanities and the sciences with fluency. The first focus of research of the Max Planck Harvard Research Center is migration and mobility in the ancient Mediterranean. Whoops. Our second focus is on discovering from the ancient biomolecules the pathogens that cause disease and massive epidemics in the human past. We've been quite successful in terms of production. Our team has made 69 major publications since 2016. You see a small selection here on this slide. So our first focus is to look at migration and mobility and to seek to reconstruct the early peopling of the ancient Mediterranean from human DNA, from the ancient DNA in the skeletons themselves. And in the course of this work, a lot of unexpected discoveries have come up. We aspired to be able to reach back to the Bronze Age, this great time of growth and change in the Mediterranean, echoed by Homer several centuries later. Would we be able to reach that in the first 5 years of operations? The answer is very emphatically yes, and we got far beyond it. Already in 2017, we were able to identify the genetic distinctions between Minoans and Mycenaeans on the island of Crete, and to shed much new light on the origins of those two populations and the civilizations that they created in the Bronze Age. In 2013, I visited the famous archaeological site of Ashkelon, being excavated by my friend and colleague, the late Larry Steger. He had just identified some graves of with grave goods marking these individuals from the Bronze Age as mysterious sea peoples. The historical records of the great Near Eastern empires record the attacks of the sea peoples in the Bronze Age, and controversy has continued for decades, if not more, about who these people were. I got Larry's skeletons together with Johannes Krause, my co-director of the MOM today, when he was an assistant professor at Tübingen. And the results were completed by our postdoc, Israeli postdoc at the Max Planck in Jena and in MOM, Michal Feldman. The ancient DNA shows that these individuals at Ashkelon shared genetic affinity most closely with people from Southern Europe. They did indeed come from across the sea. Was it from the Aegean? Were they from Cyprus? Were they from Sardinia? Stay tuned. More news, more discoveries, more analyses are coming. Other discoveries happened just because we were looking in the right place for the wrong thing. My wonderful colleague Tina Warner has identified the first woman artist of the Middle Ages to be physically identified, because in looking for traces of her diet in the dental calculus, which is a wonderful matrix for preserving biomolecules, she found these microscopic little shards of beautiful blue, which on analysis turned out to be lapis lazuli, produced in only one mine in Afghanistan. We had no idea that commercial networks connected Afghanistan to a not very significant convent in northern Germany in the 11th century. How did they get in her mouth? She must have been an artist and dipped her brush into the pigment to use this beautiful blue, extraordinarily expensive blue, more precious than gold, to cover, for example, the vestments of the Virgin in the manuscripts that this nun illuminated. Our second focus is on ancient pathogens, and particularly on one of the greatest killers of human history, bubonic plague, which is now definitively identified from the ancient DNA preserved in victims of the Black Death as of the Justinianic, and indeed as far back as far back as the Neolithic period, as bubonic plague, as Yersinia pestis. The Justinianic pandemic has been very controversial. It is now definitively and robustly identified as bubonic plague from the ancient DNA, according to the very rich but uneven written records, the plague returned between 18 and 35 times, depending on how you count. So 200 years of pandemic. That puts our 1.5 years in perspective, I must say. What were its effects? Opinions differ. The older scholarship thought that such a disease would have minimal impact on the mighty Roman Empire and the successor societies that had grown up around it. Some still maintain that. Others thought it was very important. How to determine? The written sources are insufficient. Here, this slide shows the mentions of epidemics of bubonic plague in the most important Latin historian of the 6th century, Gregory of Tours. You can see that there's a lot, but they're concentrated in one area. In fact, 9 out of 10 of them fall within 400 kilometers of his birthplace. On the other hand, you see the red triangles, which are places where our team has identified the ancient DNA of the bacterium Yersinia pestis in the skeletons of people who died from it in the 6th and 7th century. You can see there's almost no overlap between the written reports and the ancient DNA. This means that absence of written evidence of plague is not evidence of absence of plague. And it means, furthermore, that a full reconstruction of the pandemic and its changing impacts over time and space will only be possible through recovery and reconstruction of the ancient DNA. And so we've begun that task. This study was the second of our group's publications on it. We identified another 8 genomes, and you can see them in the bottom, and we did whole genome sequencing, so we got enough of the genome so that we're able to compare genomes of the individual bacteria that we've recovered from different places, from different victims. And just as with COVID, as the pathogen moves through the population, it mutates, those mutations accumulate, and so you can draw kind of a genealogy of each particular exemplar of the bug. So with bubonic plague, you can do the same thing with the DNA of the bacterium and situate each bacterium in the microevolution, constructing what's called a phylogeny, or a genealogy, if you will, a genetic genealogy of the spread, multiplication and spread of the pathogen over time and space. And now we're on the verge, and these are hypotheses that I present, but they're pretty good ones, of being able to identify individual versions, individual variants of the bacterium in individual victims with different outbreaks over the 200-year course of the disease. This means not only will it become possible to date individual burials, sometimes down to the year in which the person died a horrible and tragic death, but to spread reconstruct the spread region by region, decade by decade, perhaps even ship by ship, from place to place over 200 years. And note at the bottom, the version of the bacterium which is closest to that of the origin. The plague broke out in 541 in Egypt. The version that is closest so far to the original outbreak was found in Eastern England in an Anglo-Saxon cemetery of the middle of the 6th century. This was, for me, mind-boggling. And the implication is that a ship bearing the rats or fleas with the infection sailed directly from Alexandria or Constantinople to Britain in 541, 542, or 543, at a time when I, as a specialist of Mediterranean communications, did not believe such things were happening. Much remains to be discovered about the extent, the spread, or the lack thereof at different times in different places in order to determine the impact of the plague, of the Justinianic pandemic, in terms of economies, in terms of demography, in terms of geopolitics. It's been observed that the Arabs took 2/3 of the provinces of the Roman Empire away from it in the 600s. They came from the deserts where there were no rats. Is there a connection? The cultural impact. Art historians have observed, and historians have observed, a surge in the cult of the Virgin in the 6th and 7th centuries. Is that connected with the plague? These and many other questions are ahead of us in the science of the human past. Who are we? Where do we come from? How did we get here? The new science of the human past is beginning to uncover new answers to these ancient questions. Thank you very much.
Well, thank you. That was totally fascinating. I invite people to put their questions in the Q&A, and I'll take the prerogative to lead off. And one thing that jumped out at me, so looking at the bare outlines of your own CV with your PhD in history, you had I think, a conventional scholarly education, right? And all of a sudden, you, with your PhD in history, are talking about genome sequencing and, you know, the way a certain kind of laser investigates the air that's left in the ice. And so what strikes me is that to do the kind of meaningful work on the history of the human species that your work exemplifies, the conventional silos in which students are educated don't make sense anymore. And so I just like you to reflect a bit on, since you're also obviously you teach students today, what we need in higher education to be able to deal with the kind of interdisciplinary work, which is the work that really matters these days.
Linda, that's a great question. I was trained in the most traditional fashion in Greek and Latin philology at a very traditional university, the University of Louvain. Our program in Greek and Latin philology was started by a guy named Erasmus. So I did Greek and Latin manuscripts and inscriptions and that kind of things in the first couple of decades of my career. But I work in a period when we don't have much evidence, and I'm always on the lookout for new kinds of evidence. That's how I discovered archaeology, and then through archaeological science, the potential of DNA and other ancient biomolecules and molecules. So how do we do this? We have these tremendous disciplines that have grown up since the 19th century because of specialization. It's by specializing that you can go really deep. That's produced enormous breakthroughs across the board in history, science, philology, you name it. But as you say, that focus has turned now into silos, and we need to find ways of building bridges across the silos. And what I like to do is to introduce my students to questions. How can we figure out what happened in the health picture of the Roman Empire, and show them that we can look at text, we can look at DNA, we can look at molecules, we can look at the climate change, and how we can look at the past as a whole phenomenon, opening different windows on it from different disciplines with different toolkits. And the students respond. They love it. And they're really, really good at it. They are my inspiration. They're all of our inspiration as we go forward. They don't understand why, if you know Greek, you can't understand DNA. And I think that is where the future lies.
Okay, thank you. We do have some good questions here, and I'll start with our member, David Tatel. What new technologies do you expect in the next few years, and how do you anticipate you'll learn from them?
Wow, David, that's a great, Judge Tatel, that's a great question. I can't even guess, because I could not have guessed where we would be today 5 years ago. The ability to do whole genome sequencing on ancient DNA is new. It was only in 2015 or 2016 that somebody figured out by accident, no matter what they claim, that the petrous bone of all bones preserved DNA much better than all others. And all of a sudden we've gone from 2% success rate or 5% success rate or 10% success rate to 40% success rate. So I think that new technologies, there are improvements afoot for radiocarbon dating. New types of dating will be very, very important as established scientific dating techniques are improved. Quantitative studies of ancient texts are going to produce lots of texts that have been hiding in plain sight that no one looked at. The Bollandist fathers of Brussels have been trying to figure out which saints really existed and which didn't since 1648 and 1647. And they have published 14,000 lives of holy men and women from between about 200 CE and 1500 CE. Of those 14,000 lives, 6,000 only are dated down to about 100 years. That means there's 8,000 biographies that no one knows when they were written or where they were written. If we can figure out 10% of when and where those sources were written, we'll have 800 new documents on on antiquity and the Middle Ages. So these are going to be very, very big things. I think data science is going to be really, really big as we learn how to collect more and more measurements. We are definitely now already in the realm of big data. I had a fantastic senior thesis in which a student figured out how to collect about 500,000 archaeological records from a website and figure out a way of analyzing them to demonstrate some very fundamental changes in Roman Britain. So I think computer science, data science is going to be really big. Climate science is a relatively young discipline. It's developing very quickly. Ancient genetics is, 1980s, 1990s is the beginning of that with ancient DNA. In each of these, the technological advances have been incredible. It's really impossible to predict. They're just happening so fast. We have to keep our eyes open and our ears open and listen and talk and figure out how to integrate them in our work.
Here's a kind of maybe related question from our member Howard Block. Where and how does intellectual cultural history intersect with human physiology of the type that is now being explored?
Good question. So you saw that we now have the physical remains of the first female artist of the Middle Ages. She produced manuscripts. She produced work. Nothing survives from her convent. Is there a manuscript of her still around? There may be, and maybe her ancient DNA is on it from the saliva that she put on her brush when she put on that manuscript. We are able to reconstruct the details of this woman's personal existence, her diet, her health crises, her age at death, her family networks. All of these things are things that we can know now thanks to the science of the human past. So if we study the beautiful things that human beings have created in terms of literary works, in terms of philosophical systems, in terms of artworks, the Parthenon, the stele of the Keramikos cemetery in classical Athens, these are magnificent things that we've used as windows into the people. But the people who created them now for the first time are becoming accessible to us in their biological historical existence. And through understanding the people themselves, their relations, their diets, their challenges, physical and other, we will understand far better the artifacts of mind and of hand that they produced in the course of their brief existences.
This is more of a comment than a question, but it may ask you to expand on what you said in response to my question. From Caroline Bruzelius. She says, picking up on Linda's comment, this kind of groundbreaking work requires research by collaborative teams, a type of work that strains our organizational and administrative structures of universities, not to mention evaluations for promotion and tenure within the humanities departments. I could see that as a problem. So I guess the question really is how to, administratively, how to break through the built-in siloization of what it takes to succeed in conventional academia.
By having a set of armor and smashing yourself into the silo over and over and over again until there are enough colleagues smashing themselves with you that you break through the silo. It's a tremendous challenge, Caroline. Really difficult. It's a challenge on every level. It's a challenge of just getting people involved. It's a challenge of helping people understand. You have to make a considerable investment to understand. You don't have to be able to do the complex mathematics of the genetic analyses yourself, but you have to be able to understand them and judge whether they make sense or not. And that's a huge investment. The administrations are stacked against us. They constantly say they want interdisciplinary, transdisciplinary work. And when you present a study of disease using DNA in a history department, you're dead. No one's going to finance that from a historian's point of view. Getting the physical resources to do this work is incredibly difficult, as exciting as the results are. So the intellectual, the inertial challenges are enormous. I find the concern that multi-authored publications, which might be part of your question, will complicate the rise to tenure for junior professors in the humanities a false dilemma. Great universities are deciding every day on tenure for dozens of beautiful, wonderful young scientists who are lead authors in multi-authored publications. And scientists know pretty darn well. The Nobel Prize Committee does a pretty good job of figuring out who in the 50 people on that signature deserves the Nobel Prize. So it's a little bit off-putting if when you first get started, but our scientist colleagues are doing it every day, so it shouldn't be a major problem beyond kind of cultural comfort in terms of teaching promotion committees to appraise the work of individual young scientists leading these research projects.
Sadly, that has to be our last question. We have to wrap up, but I am here to tell you that you'll be back speaking to us, I hope, in person as this kind of work goes forward. So thank you very much.